Hi, my name is Maciej Torak, and today I will present executable biochemical space for specification and analysis of biochemical systems. I will start with motivation. In systems and synthetic biology, there are very frequent issues with mathematical modeling. One of them is reconstruction, which is an action of extracting information from data and previous models, and then it's understanding it with the relevance to the biological meaning. And finally, with all this information together, we need to interpret the model or the results of analysis. To help and target these issues, we are developing so-called comprehensive modeling platform. This platform consists of several modules. One of them is modeling part, another is experimental part, and these parts are connected together in so-called biochemical space. This is a central part, and today we will talk mostly about this part, and also we will focus on description part of this module. Just to mention, there are already two instances of comprehensive modeling platform. It's Cyanobacterium for modeling of cyanobacteria processes and photosynthesis, focusing on photosynthetic processes. So biochemical space, it's a semi-formal knowledge base which provides specification and annotation. The specification is given in a formal language and helps us to define objects and relationships among them. Then annotation helps us to assign a meaning to the objects. In this case, it's a biological meaning from the particular domain. Here is example of a record from biochemical space. As we can see, there is specification and annotation part. The specification tells us how exactly some objects are interacting. This is formalized in a language, while the annotation tells us some additional information about the objects and assigns them the biological meaning. This is done in particular by relating them to some biochemical databases such as CAC, Protein Data Bank, UniProt, and so on. The specification part is defined in so-called biochemical space language. Why this language in particular is because it's important to note that the target group of users is outside of computer science. It's mostly experts from biology, mathematics, or chemistry. Therefore, the biochemical space language combines and focuses on following aspects. Human readability. It's important such that the notation the language is using is really easy to read, write, and maintain. Then it's using so-called rule-based approach, which helps to avoid combinatorial explosion on the level of description. Also, such as all the other rule-based languages, it's using its own level of abstraction. Then the language uses some sort of hierarchy which allows to assemble more complicated objects from simpler ones. And finally, a crucial aspect of the language is that it has operational semantics, which allows us to perform additional analysis on the models. Here is a schematic example of hierarchy of agents. I will talk about each class in more detail, but it's important to understand that there are some sort of atomic agents, which are the simplest ones. Then there are structure agents, which are composed from atomic agents. And finally, there are complexes, which connect together structure and atomic agents. And finally, each complex has assigned a so-called compartment. So atomic agents describe the most simple objects. They have assigned an internal state, and this state can be changed. When we are modeling a system, basically atomic agents are crucial in the level of abstraction we choose. We can, for example, model changes on atomic level. So as an example, we can see a cytosine methylation, and also it's possible, for example, to model a phosphorylation of a protein. Here we have examples how it can be written in a biochemical space language. Structure agents, they have a name and assigned a set of atomic agents. A typical representative is a protein with amino acids residues, and these can have assigned some states. So for example, we have a protein with serine phosphorylated residue and cytosine unphosphorylated residue. Finally, there is complex agent, which represents coexistence of atomic or structure agents. It is important to note that this is really a general coexistence. It doesn't express any particular order or any binding information. So we can model, for example, a protein complex, but also a solution of water and salt, for example. And this complex has assigned a compartment which determines special position and can be, for example, used for modeling of mass transport. All these agents are used in a rule. A rule describes a pattern according to which agents can be changed. It's important that it's a generalized version of chemical reaction, and we can basically omit all the context which is not important. In this example, we have again a protein with 2 amino acids, and each of them can be phosphorylated independently. So for that, we can write 2 independent rules. In the first one, the serine is phosphorylated, and in the second one, the threonine is phosphorylated. This way we can focus on a particular substructure of modeled issue. Here are some more examples of rules. We can generalize them to some categories such as state change. We can change the state of a particle atomic agent or atomic agent in a complex or inside a structure agent. Then there is complex formation, which is also a typical interaction. And finally, there is a transport. We can, for example, relocate a protein to a different compartment, or we can define also rules which model some boundaries of the system. Also note that we can assign a rate function to which defines how likely is the rule to be applied. As I mentioned in the beginning, it's important that the language has also operational semantics, so we can perform some analysis. So we can then, for example, generate a transition system and perform model checking or parameter synthesis. What we are also trying to do is focus on static analysis. However, static analysis in rule-based systems is not well established, not as good as in Petrinets or reaction networks. However, it's really worth exploring this area. Finally, we have developed software support for this language. It's available online here, and it has features such as interactive editor or stochastic or deterministic simulations. Also, it's possible to perform parameter synthesis and model checking for probabilistic CTL. And of course, there are multiple interactive visualizations, for example, for time series or for exploration of transition systems, and also for visualization of PCTL model checking and parameter synthesis results. Finally, there are several static analysis methods implemented which can find some inconsistencies in models or some redundant rules, for example. To conclude, we have presented biochemical space language, which is used to basically formalize biological knowledge and assign it or integrate it with annotation information. As future work, we want to focus more on static analysis, and also we would like to work on SBML multi-compatibility, which is a SBML package for support of role-based languages. Thank you for your attention.